So what my goal is for today is to finish reviewing bacterial pathogens, maybe talk a little bit about viruses. We're also going to talk about fungi. So we're going to start with the infections of digestive system. So it's going to be, let's call them enteric pathogens. Okay. So what can we put in this group? First of all, it is, of course, Enterobacteriaceae, the enteric commensals, so there are 4 species that I specifically put in this group. It's Enterobacter aerogenes, Pteus mirabilis, Klebsiella pneumonia, and Serratia marcescens. I'm not sure about the spelling of marcescens, but it's fine. So, few things about them. They all are nosocomially transmitted, which means that they are transmitted in the hospital settings. And since they are transmitted in the hospital settings, we can consider them being opportunistic infections. The most frequent types of opportunistic infections that commensal Enterobacteriaceae can cause are wound infections, pneumonias, and UTI, urinary tract infections. So if you look at this, sorry, this list here, you may notice that what does commensal mean? It means that these microorganisms can be found in the human gut. Okay, so they basically come from the human gut and cause infections as a result of this. We're good so far? Okay, good? Okay, all right. Next, technically, it is also a commensal. I just put it separately. It's E. coli. Okay, so E. coli is another intestinal microorganism that can cause, most of them cause diarrhea, urinary tract infections. It's one of the main causes of neonatal meningitis. Now, transmission can be as well as for Enterobacteriaceae, can be nosocomial transmission. Now, one particular strain that I wanted to address here, I have a whole bunch of strains of E. coli in the lecture notes, but one that I really want to address is enterohemorrhagic E. coli, EHEC. So enterohemorrhagic E. coli causes dysentery, which is basically a blood stool. Okay, I think it is. Hello, Mary. So we're going to figure out the exam thing, okay? Don't worry, you're going to take your exam, no problem, okay? We'll just figure out the time. Sounds good? Can you hear me? Yeah, we'll figure it out. So we're going to do the review, and then when we're done with the review, just stay on WebEx, and we'll figure this out. Sounds okay? Okay. So let me go. So EHEC can cause dysentery and so-called hemolytic uremic syndrome. Sorry, the wrong letter. So what is dysentery and what is hemolytic uremic syndrome? Dysentery is the bloody stool because infection causes the lesions in the colonic epithelium. And this microorganism ends up in the circulation, causes the breakdown of red blood cells. And breakdown of red blood cells responsible for hemolysis. So the result of this breakdown, kidneys start to shut down. So that leads to uremia, and therefore, altogether, it's the hemolytic uremic syndrome. Now, this particular virulence of EHEC is due to the presence of Shigella toxins that inhibit protein synthesis. And the transmission route for EHEC is food. That's the primary transmission route for this microorganism. Mostly the ground beef part of the food. Now let's talk about some more virulent microorganisms. And we're going to start with the Salmonella here. So Salmonella is a foodborne infection which is essentially a zoonosis. It is acquired from animals. Well, not directly. It can be acquired directly from animals, but humans usually acquire Salmonella through the consumption of contaminated milk, poultry, or eggs. Salmonella can cause enteritis. It's kind of a mild form with vomiting and diarrhea. Or more severe form of Salmonella is typhoid fever. Which is characterized by, well, really high fever, multi-organ involvement. Now, sorry. Fairly close to Salmonella in terms of the manifestations is Shigella. So Shigella is another foodborne zoonotic infection. But Shigella is more severe than Salmonella since it causes dysentery. Due to the presence of, well, just like with EHEC, Shigella toxins. Which, as I mentioned before, inhibit protein synthesis. So far, we're good? Okay. So the last group in enteric pathogens that I wanted to discuss here will be Vibrio. And I'm going to start with the enteric Vibrio cholerae. So Vibrio cholerae is, I'm going to put a little arrow here. Vibrio cholerae is halophilic microorganism. Not too halophilic, but it prefers brackish water. It is a waterborne pathogen, so it is transmitted when people drink contaminated water, and it causes profuse diarrhea. The toxin stimulates production of cyclic AMP, and that those elevated levels of cyclic AMP leads to the increased secretion in the intestine, and that what leads to diarrhea. Now, interesting thing about Vibrio cholerae is that it is often found on the small crustaceans really microscopic crustaceans in the water. And if you filter those crustaceans out, for instance, by passing water or filtering water through 4 layers of cloth, you can reduce the bacterial burden in the water by basically several orders of magnitude. Another member of the Vibrio genus that I wanted to really briefly mention, it's not a digestive microorganism, it's Vibrio Volificus, which can be acquired from water or shellfish and causes pretty severe wound infections. So untreated Vibrio Volificus, of course it has to have a wound to get into, and the severe Vibrio Volificus can be pretty dangerous and even lethal. Before we move any further, I want to ask if you have questions, and I want to ask if I can kill this stuff on the board. I'm asking everybody. You can just say like, yeah, sure, like fine. We good? I'm looking this way because my camera is here, but the screen where I see everybody is here. It's kind of, different. We good? Okay. So now we're going to talk about a few microorganisms which can be considered severe bacterial zoonosis. So we're actually going to call them this way. So among severe zoonosis, the first genus that we're going to talk is going to be the genus Yersinia. And of course, in the Yersinia genus, there are 2 species I wanted to mention. One is pretty mild. It is Yersinia Enterocolitica, which is, well, zoonotic infection. And it can be acquired from dogs and cats that are not entirely housebound and kind of semi-feral, can walk around, And it causes gastroenteritis. Yersinia Enterocolitica is an example of an orphan disease. Doesn't mean that it affects orphans. It means that it is often overlooked by physicians and is not considered as being an actual cause of a particular case of gastroenteritis. Of course, the second species that I wanted to mention here is Yersinia pestis, zoonotic agent that causes plague. Okay, so plague generally can be subdivided into 4 categories: bubonic, pneumonic, and septicemic. So bubonic is the sort of easiest one. Pneumonic is much more severe. So the mortality rate for bubonic plague is around 70%, for pneumonic is around 90%, and for septicemic is around 100%. So the transmission route, That's the wrong letter. Fleas can transmit this infection to humans from animals. Flea takes a bite on an infected animal and then bites a human and transmits the infection to the human. Between the humans, it can be transmitted by respiratory route or contact between humans. So how does infection occur? If a person is bitten by an infected flea, microorganism migrates to the closest lymph node, leads to the necrosis of the lymph node, and that is called a bubo. If this person can develop septicemic form or pneumonic form, the infection can be transmitted from person to person via respiratory or close contact. There are cases of plague in the United States every year. It's absolutely normal. We do have plague in the US. And another severe zoonosis that I wanted to mention here will be Francisella tularensis, causative agent of a condition called tularemia, which can in two forms: cutaneous form and respiratory. So it is a zoonotic infection which can be acquired from rabbits. It's pretty severe. In its cutaneous form, though, it's relatively mild and usually acquired when people skin infected rabbits. In its respiratory form, it can be very severe with the mortality rates up to 50%. It is not transmitted from person to person, but very effectively transmitted via aerosols when rabbit droppings are aerosolized and whatnot. Any questions on this? Any questions? No? William, Emily, we're good? Moving on? Okay, awesome. So now, three species that I wanted to mention in our next installment cannot really be grouped in any particular way. We're going to start with Pseudomonas Iroginosa. Which is quite an interesting and fascinating microorganism. It is found in the environment. That can be transmitted nosocomially. So it's a hospital-acquired pathogen. It is a typical opportunistic pathogen, which can cause a variety of skin infections, urinary tract infections, but it is most famous for causing pneumonia in the patients with cystic fibrosis. Okay, particularly in patients with cystic fibrosis. Another feature of Pseudomonas is that it is notoriously antibiotic resistant. Very much antibiotic resistant. Next one on our list is Legionella Pneumothorax. So this microorganism can be acquired from the environment, and I will specify a little bit more on how it can be acquired from the environment. What's interesting, it as you can guess from the name, pneumophila, it causes pneumonia. But this pneumonia is an example of non-communicable infection, meaning that a person with legionellosis cannot transmit the infection to somebody else. Now, this non-communicable pneumonia is caused because this microorganism infects amoeba in nature, but it infects alveolar macrophages in humans. So this bacteria normally in the environment, it is a pathogen of unicellular protozoa, amoeba, which reside in water. When people inhale water aerosols and Legionella and infected amoebae end up in the human lungs, Legionella can infect alveolar macrophages and survive in macrophages and cause lung damage, basically pneumonia. So far, we good? Does that make sense? Good? Okay. So, third microorganism on that improvised list here is Haemophilus influenza. Haemophilus influenza is a respiratory transmitted human pathogen, and it can cause pharyngitis, which is less frequent, because it can cause otitis media, so infection of an inner ear. But it is most common, most known for community acquired meningitis. Along with Streptococcus pneumonia and Neisseria meningitidis, Haemophilus influenza is the third most common cause of the community-acquired meningitis. And I want to stress it out. Haemophilus influenza has nothing to do with actual flu, with influenza. So far, we good? Okay. Thankfully, there is a vaccine against this infection. And the last microorganism that I'm going to put on that particular, that set, before we move on to our D-Derms, is Helicobacter pylori, which turns out to be most likely a human disease, so human-to-human transmission. It is in the stomach. Can be actually a part of the stomach microbiome. So as surprising as it is, Helicobacter pylori doesn't always cause, but it is a cause of peptic ulcer. This microorganism possesses enzymes like urease that neutralizes acid to protect microorganism from being killed, and mucinase that breaks down mucus and leads to the damage to the epithelium and peptic ulcer. One of the main reasons we know that peptic ulcer is the infectious disease is because we can treat it with antibiotics and cure it with antibiotics. Before we move any further, we good? Any questions? Fine? All right. We have a last, and yes, I do record this session. And it's probably going to be really decent for you. So our last group of bacteria that I wanted to talk about are D-Derms. Now, D-Derms are not considered to be either gram-positive or gram-negative. They their own rule, and they are all spirochetes. We're going to actually name them D-Derm spirochetes. So all these three microorganisms are spirally shaped. They're relatively thin. That's why they are best observed using the dark field microscopy. So first microorganism in that list is Leptospirosis interrogans, which is a zoonotic cause of the condition known as leptospirosis. So zoonotic transmission occurs when a person gets in the contact with the feces or urine of mostly infected dogs. And leptospirosis by itself is, I wouldn't say mild, but it's a typical bacterial infection with garden variety fever and muscle and joint aches. But in some cases, particularly in the people who are immunocompromised, it can progress to so-called whale syndrome, which is much more severe and can be characterized by the kidney failure, liver failure, and splenomegaly. Another D-Derm that I wanted to mention is Borrelia burgdorferi. A causative agent, zoonotic causative agent, causing Lyme disease. It is transmitted by ticks, which are example of biological vector here. And Lyme disease manifests itself usually as myalgia and arthralgia, but if it's not treated in a timely manner, it can progress to chronic arthritis, paralysis, and myocarditis. One of the really, really distinguishing signs of tick borreliosis is so-called bullseye rash. Rash that looks like this. So you've got a rash in the center, and you've got in the periphery, you've got rash as well, like a circle around. It's called bullseye rash or erythema nodosum. Erythema migrans. Erythema nodosum is a different thing. Okay, we're good so far? Lastly, the most exciting, if we can say this, of them all is microorganism called Treponema pallidum, a causative agent of syphilis, Begel or yaws. Begel and yaws are sort of milder forms of syphilis, and it is, of course, a sexually transmitted disease. The question often arises, can syphilis be transmitted from person to person by contact? The answer is not really. There were cases of human-to-human transmission of syphilis, and people shared pretty intimate objects such as towels or even underwear. So yes, that can happen, but by contact, like kissing or hugging or holding the hand, no, syphilis cannot be transmitted. There are 3 stages for syphilis: primary stage, secondary stage, and tertiary stage. Only primary and secondary are infectious. In the primary stage of syphilis, the symptoms usually include so-called chancre on the mucosal membranes of the reproductive system. Secondary stage is characterized by the rash on the mucous and cutaneous membranes, low-grade fever, multiple lesions to the reproductive organs. In tertiary, syphilis microorganism becomes systemic and affects different organs such as heart, nervous system, blood vessels, connective tissues. I don't have any images of tertiary syphilis in my lecture notes because those images, quite frankly, are pretty graphic. You can Google it yourself. It's bad. Any questions so far? Okay, if not, then we will move on and we will talk about fungi. Okay, so fungi can be divided into kind of three groups. First will be molds. Second group will be yeast. And the third, relatively small one, will be dimorphic fungi. Let's start our conversation with the molds. So molds are multicellular fungi. They consist of septate or non-septate hyphae. So what are septate or non-septate hyphae? If individual fungal cells remain individual, we call them septate. There are cell walls between the cells. If cells fuse, we call such hyphae non-septate. Hyphae form mycelium. When you find mold growing on the bread or berries or fruits, this fuzzy structure that you see is a mycelium. Inside of the mycelium, you can find vegetative or reproductive hyphae. So vegetative hyphae are essential for acquiring nutrients from the substrate the fungus is growing on. So if you look at the mold growing on the strawberry, vegetative hyphae will be in the strawberry. Reproductive hyphae, as you can guess, responsible for reproduction. And probably what you see on top are reproductive hyphae. So what are the reproductive strategies for molds? Hyphal fragmentation, and two types of spores: asexual spores and sexual spores. So hyphal fragmentation is just when hyphal fragments fly away without forming spores. Pretty much like a tree branch, sometimes, if it's freshly snapped, can be placed in soil and eventually give rise to a new tree, a new trunk. Now, we're going to talk about difference between sexual and asexual spores in a few minutes when we get to the yeast. Yeast are unicellular fungi that can form, however, pseudohyphae. So what is pseudohyphae? Pseudo. I misspelled it a little bit. Pseudohyphae. So yeasts can reproduce by budding, which is essentially an example of mitosis. When a yeast cell divides, new cell often does not separate from the mother cell, and they start forming a chain of individual independent cells. That chain of individual independent yeast cells is called pseudohyphae. Other strategies for the yeast reproduction include also asexual and sexual spores. So in both cases, for both yeast and molds, asexual spores, they are produced, that's basically normal way. They are produced frequently, no problem, all the time. Now, what about sexual spores? So sexual spores are produced under environmental stress. Okay? So sexual spores are produced under environmental stress. They do contribute to some extent to the genetic diversity of fungi, but they are by no means the main way of fungal reproduction. Asexual spores are the main way. Asexual spores are the main way. Sexual spores, not. Now, what are dimorphic fungi? Dimorphic fungi are the ones that grow as molds in nature, yeasts in humans. We already encountered the switch for changing growth modality is most likely temperature. Because humans, inside of the human body, it is much warmer than in the ambient environment. Now, a few words about spores. I want to focus particularly on the asexual spores, but both for yeasts and for molds. The asexual spores can be divided into conidiospores and sporangiospores. So what is the difference between conidiospores and sporangiospores? The difference is structural. For sporangiospores, you can look at it this way. You have a sac called sporangium, and inside of this sac you have a whole bunch of spores. When this sac bursts, spores fly out. In case of the conidiospores, you have individual spores that form a chain, and they separate one after another. So there's no sporangium. Does that make sense? Are you following me? You good? Good? Fine, okay. Any questions? All right, so we're going to talk a little bit about antifungal drugs, and then we're going to review, well, fungi. So antifungals, these include three main categories. The first group of antifungals is polyene drugs. And again, you need to know only 4 classes, not any specific brand names or drug names. So polyenes, which include nystatin and amphotericin B, they disrupt cell membranes. Now, as you can imagine, since fungi and humans are both eukaryotes, antifungals in general have a pretty considerable level of side effects. And indeed, nystatin and amphotericin B are pretty toxic drugs. Second class, quote unquote, of drugs is griseofulvin. So griseofulvin inhibits microtubule synthesis. Microtubules are essential for the cell division, so griseofulvin essentially controls and prevents fungal cells from dividing. Now, next category of antifungals is a drug called flucytosine. I think it's a little bit too much of a chicken scratch. Which basically is a nucleoside analog allowing us to inhibit DNA synthesis. And finally, the fourth class of drugs here are azole drugs. So azole drugs inhibit ergosterol synthesis. Azole drugs are by far the best antifungals at our disposal, such as miconazole, ketoconazole, fluconazole. Ergosterol is the key molecule in the membrane of fungal cells. The inhibition of its synthesis essentially disrupts the structure of fungal but not human membranes. Any questions so far? Good? Okay. Can I get rid of this? My concern, let me explain why I'm asking this. My concern, like, are you catching up? Okay, good. I'm trying to pace myself fairly well. So that brings us to a brief review of different mycoses. And we're going to start with kind of cutaneous mycosis. So cutaneous mycosis, there are three categories of pathogens that I want to focus your attention at. So first group of pathogens are the ones that cause ringworm. This group will include trichophyton, epidermophyton, microsporum, and Moloscesium. So these four genera, they cause a variety of skin fungal infections. And I'm gonna, so skin infections, skin fungal infections. For example, ringworm. Now, ringworm, you're all familiar, like on the head, right? Or on the hands, athlete's foot. A jock's itch, and many more. Basically, all these microorganisms infect epidermis and lead to like inflammation of epidermis. They can affect practically everybody, can be transmitted from person to person, from animal to person. I really want to stress this out. These infections, although they can affect healthy, not healthy people, doesn't matter. They are restricted to the skin. They do not get any further. And in general, fungal infections of humans, they are superficial and they are opportunistic. I'm going to actually put this statement here at the bottom. Fungal infections if systemic affect immunocompromised individuals. So what does that mean? Given that you all and I are healthy adults, our chances to develop truly a systemic fungal infection, like respiratory fungal infection, respiratory mycosis, or neurotropic mycosis, our chances are extremely low. Fungal infections are opportunistic and affect, first and foremost, immunocompromised individuals. Skin is the different story, because what it is most likely, fungi don't like to be very warm. Human body inside is pretty warm, but in the skin, it's kind of okay. So the thought is, this is why people easily develop cutaneous fungal infections, but not systemic. So far, any questions? Okay. Another genus is, well, not even genus, Candida albicans, a species of yeast that can cause infection of the nails, okay, and mucosal candidiasis, which can manifest itself as the thrush. You're all familiar with this oral candidiasis or fungal vaginosis, so vaginal candidiasis. Again, look, infection is restricted to the superficial layers of the human body, either almost external mucosa, or infection of the nails. And finally, the third kind of interesting group is Aspergillus, which causes cutaneous infection. It is usually secondary to systemic aspergillosis. So let me explain what it means. If you have an immunocompromised patient, who has a systemic infection with Aspergillus, the microorganism can travel by the means of the blood and end up somewhere close to the skin and cause skin infection from within. Does that make sense? Okay. So our next stop in conversation about microorganisms, the fungal infections, is neurotropic mycosis. And in this group, I'm going to talk about two pathogens. One of them is Cryptococcus neoformans. Sorry, there's a massive typo here. Causative agent of meningitis. Now, this meningitis affects mostly AIDS patients. It's so-called hallmark infection of the AIDS patients. Just like any other meningitis, it is characterized by the stiff neck, high fever, confusion, headaches. Now, the virulence factor that contributes to this infection being really hard to treat is a capsule, which protects cryptococcal cells from phagocytosis. The infection is zoonotic. Can be acquired from bird droppings. The second species is Xidioides immitis. Xidioides immitis can cause meningitis as well. Can be acquired from the environment. Pretty common in southwest United States, like California and Nevada. Affects mostly immunocompromised. And it's also highly contagious. Now, I want to highlight this neurotropic mycosis, and we're going to talk about respiratory mycosis a bit later. Those are non-communicable infections. Once it's in the CNS, that's it. Now, both Cryptococcus neoformans and Coccidioides immitis are acquired by inhalation of spores. Okay, so it all starts in the lungs and then moves into the brain. So far, does that make sense? Okay. Now, next, basically, the last group of fungal infections that I wanted to address here are respiratory mycosis. Now, again, this division is very, very much artificial. Okay, you will see it in a second. So first, practically truly respiratory mycosis is Pneumocystis jirovecii. It's the fungal infection that causes typical pneumonia, which affects mostly AIDS patients. Okay? It is another hallmark disease of AIDS patients. So yeah, typical symptoms: shortness of breath, non-productive cough, low-grade fever. Another name, just to let you know, you may encounter this acronym. It's sometimes called PCP, which stems from the old term. It used to be not Pneumocystis jirovecii, but Pneumocystis carini. So Pneumocystis carini pneumonia. But this is why people don't like taxonomists. Genus Blastomyces is another example of fungi that are hard to characterize because it can cause skin infections as well as pneumonia, but it affects mostly immunocompromised patients. And finally, the third microorganism that I wanted to highlight, you already heard about it today. It's Aspergillus. This fungal genus grows as a mold in the environment and can actually grow as a mold in humans. When spores of Aspergillus, an example of Aspergillus is the black mold. You've all heard about it, I guess. When fungal spores end up in the lungs of immunocompromised patients, they can form so-called hyphal bulbs. You've all seen hyphae, you've all seen mycelium, molds growing on things like bread or strawberries or fruits. Imagine that this same growth, the ball of fungus, but in the human lung. Respiratory aspergillosis is a really, really severe disease. All of these infections are acquired from the environment, actually very characteristic for fungal infections to be acquired from the environment. Any questions so far? Good? Good? Okay. Questions, Mary? No? Good? Emily, Dolomian, you all catching up? Okay, so we're going to move on, and we're going to talk about viruses. And we're going to do a brief review of virus structure, replication strategies, and so on. So let's start with classification. So viruses they have a couple things that are common. They are obligate intracellular pathogens. And why? All viruses have no ribosomes and no own metabolism. So they have to use cellular machinery to produce proteins, and they have to use cellular sources of ATP. Structurally, viruses can be divided into naked viruses or enveloped viruses. So naked viruses can be pictured the following way. Viral genome is enclosed into the viral capsid, which consists of proteins. Now, naked enveloped viruses also have a genome and the capsid. But on the outside of the genome and the capsid, you can find an envelope which consists of proteins and phospholipids. Okay? So far, are we good? So you kind of see. To clear my conscience, I must say that there are viruses that have no capsid, but we are not talking about them. They are not human pathogens, or at least we don't know any human pathogens. Now, what about the viral genome? Since viruses, as you can see, have no cellular structure, that are not considered to be alive. And you know that all living things have double-stranded DNA genome. Viruses do not have that restriction. The genome can be DNA or RNA. Okay? It can be both double-stranded DNA, like herpes, or single-stranded DNA, like parvoviruses. And same is true for RNA viruses. They can be double-stranded RNA genome, like rotavirus, or single-stranded RNA genome. And within the single-stranded RNA, there is a further breakdown. Single-stranded RNA can be positive and negative. So this concept requires some further explanation. So if you remember the story about, sorry, I have a brain fart, story about the gene expression. Remember, double-stranded DNA is transcribed into single-stranded RNA, and single-stranded RNA can then be translated into protein. So by definition, if single-stranded RNA can be used as the template for protein synthesis, that is considered to be positive. So positive single-stranded RNA can be immediately used for protein synthesis. Negative single-stranded RNA has to be complementary copied into a positive form to start protein synthesis. Does that make sense? I usually compare this to like a photograph, and this is a negative that has to be converted into a photograph. So far, we good? Questions? Okay. So structurally, viral capsids can be broadly divided to three main categories. Helical capsids, this is the self-explanatory, okay? Helical, well. icosahedral. I'm not particularly good at 3D geometry. Think of it as 12 triangles assembled in something that looks like a sphere. Okay? This is perhaps the most common form of viral capsids, icosahedral. And finally, complex capsids. Complex is the category which kind of has all the viruses with some unconventional capsids like filamentous capsids or bullet-shaped capsids like for rabies, or without any particular description like for pox viruses. Into that category also fall the capsid of a so-called T-even bacteriophage. So you may have seen this image before. Okay, so that's your typical T-even bacteriophage, the capsid on top, the tube covered with a tube sheath, the tail fibers, and the tail pins. Okay, that's a classic complex capsid. They do look like this. They do look like tiny alien spaceships. Now, this actually provides us with a good segue into the conversation about bacteriophages. So let's talk about two types of bacteriophage infection. And I mean, unfortunately, well, you don't have that convenience that I have to draw and erase, so I will have to do this. I will try to pace myself decently. So, bacteriophages, and I will try to use the entire board here. They can infect only bacteria and archaea. So there are two main types of bacteriophages. Lytic bacteriophages and lysogenic bacteriophages. So I'm going to explain the differences in replication cycle for both of them. And I'm going to start with the lytic bacteriophage. So here you can see a bacterial cell. I will actually make it a little bit smaller. And I'm going to use this color for everything bacteriophage. I'm not going to draw any specific like this structure. I'm just going to use the circle with the DNA in it. So, bacteriophage attaches to the surface of the cell. That's the first step of infection, called attachment. And then it injects its genome into the cell. This process of injection, and I want to focus your attention on the genome. Capsid does not get into the cell, okay? Only genome. So that is penetration. Inside of the cell, genes encoded by this genome get expressed, which leads to the production of bacteriophage proteins and accumulation of bacteriophage genomes. The third step is called biosynthesis. After proteins are produced and genomes are copied, bacteriophage assembles and its particles are released from the cell, leaving holes in the cell wall, basically. Now, this process is called egress. Egress, release, exit, all those names are appropriate. Now, you can imagine that when bacteriophage is packaging its DNA, it can accidentally grab a fragment of the bacterial DNA instead, and in this case, forming something like that. Okay, instead of grabbing its own proper DNA, it grabs bacterial DNA. This is called generalized transduction, the way to move the genetic information from one bacterial cell to another. Now, we call this bacteriophages lytic because they replicate once they get into the cell, and cell immediately, well, almost not immediately, in a very short period of time, it dies. Okay, it gets destroyed. Phage DNA does not get incorporated into bacterial chromosome. In case of lysogenic bacteriophages, the situation is slightly different. So the phage just like lytic phage gets attached to the bacterial cell, injects its DNA inside of the cell, and here you have bacterial chromosome. But then quite an interesting thing happens. So phage DNA gets incorporated, becoming a part of the bacterial chromosome. Can you see that? Can you see that? It becomes a part. So this phage DNA inside of the bacterial chromosome is called a prophage. And every time bacterial cell divides, it copies the chromosome and it copies the prophage with it. So virus basically wastes no energy. It does not spend any resources on spreading throughout the bacterial population. Now, this phage being stuck in the bacterial chromosome at some point can be excised. That's your tiny little phage here, okay? So, and the stimulus for that excision is environmental stress. Genes are being expressed. Which leads to the assembly of phage particles and their release from the cell in the environment. Does that make sense? So as you can see in this case, phage infection also results in lysis, but this lysis is postponed. So the difference between the lytic and lysogenic bacteriophage basically is that in the case of lysogenic bacteriophages, bacteriophage DNA gets incorporated into the bacterial chromosome, and the cell death, the lysis, is postponed. I hope that makes sense. Good? Good? That makes sense? All right. So let's review the typical infection of the animal virus. I'm not going to title it anyway, in any particular way. I'm going to, though, make a gigantic, well, not too gigantic, a eukaryotic cell with a nucleus and let's do it. So first, virus. I'm going to use just garden variety, non-envelope, naked virus. Has to bind to its receptor. So just like in a case of bacteriophage infection, first step is attachment. Next, the whole virus has to get into the cell. It can be achieved by the fusion or endocytosis. Let's look at endocytosis. So that's endosome. And inside of that endosome is a virus particle. So that step is called penetration. You see the difference between the animal virus and bacteriophage. Bacteriophages, in case of bacteriophage capsid, doesn't get into the cell. In case of animal virus, it does. Due to the usually acidic conditions in the endosome, capsid starts to fall apart. So that leads to this little virus fragments of capsid, and the release of viral genome. This step right here, third step, is called uncoding. Okay? Uncoding step is not present in the course of bacteriophage infection. So once the genome is released, there are two ways. One, genome can end up in the nucleus, or it can stay in the cytoplasm. Usually, and I don't know how to stress it enough, usually DNA viruses, the genomes end up in the nucleus. For RNA viruses, the genomes remain in the cytoplasm. Okay? In both cases, whether in the nucleus or in the cytoplasm, there is a step of biosynthesis. That's number 4, right? So biosynthesis results in the accumulation of individual nucleic acid and proteins, which then assemble into a mature virus. So it's number 5. And after assembly, the virus is released from the cell. So I'm going to go over it one more time. Virus can attach to the receptor, has to attach to the receptor. Virus particle gets into the cell via endocytosis or fusion. Viral genome undergoes uncoding. It is liberated from the virus particle. And then for most DNA viruses, genome ends up in the nucleus. For RNA viruses, it stays in the cytoplasm. Genes getting expressed, producing viral proteins. Viral genome is copied. Viral genomes and proteins assemble into the mature particle, particle leaves the cell. Few sort of details. First, why DNA go in the nucleus, RNA in the cytoplasm? DNA in the nucleus is normal. DNA in the cytoplasm is not. So DNA viruses essentially try to hide the DNA from cellular pattern recognition receptors. Another thing, they may use the machinery, the DNA synthesis machinery that is found in the nucleus. Okay, so that may contribute as well. Second, I want to say a few words about the presence or absence of the receptor. So if the cell if there is a receptor present on the cells are called susceptible. For instance, human cells are susceptible to infection with a poliovirus. Mouse cells do not have the receptor for poliovirus, so they're not susceptible. If the virus can replicate, cells are called permissive. So same example, human cells are permissive for poliovirus replication. Interestingly enough, mouse cells are permissive too. If you can somehow stick the virus into a mouse cell, it will replicate in there, but it can't get in. So the virus can get into the cell if cell is susceptible, if it has a receptor, and it can replicate in the cell if the cell is permissive. So for a successful infection, cells have to be both susceptible and permissive. We good so far? Okay, before we move on, any questions? Good? That's actually, I think it's going to be like the best recording that I've made for this one so far. So I will upload this on the YouTube. Right. So I'm going to briefly review some viruses with you, if you don't mind, okay? So we're going to start with the one that you should have gotten vaccine for. I hope you all did. Okay? Let me think if I can actually, how about this? I'm going to try and group. No, tough luck. I basically talk about positive single-stranded RNA viruses. Okay, let's talk about influenza first. So influenza is a single-stranded RNA virus. RNA is negative, and genome is segmented, meaning that you have not one molecule, but 8 individual single-strand RNA fragments. It's enveloped virus. Okay. And 2 key proteins found in the envelope is hemagglutinin H, which is responsible for receptor binding, and another one is neuraminidase. Which is an enzyme essential for infection and release or egress. Okay? So this there are many types of hemagglutinins and many types of neuraminidase, which allows you to classify influenza based on these two proteins into something like H1N1, H3N2, etc. Only certain combinations are typical for human infections. Okay? So what about transmission? It's a zoonotic disease. So there are several ways, and we're going to try and picture it nicely. It all starts in waterfowl. Then it is transmitted, and in waterfowl, influenza is GI infection. Okay, so in waterfowl, influenza infects digestive system. From waterfowl, it gets into pigs. And in pigs, it's upper respiratory tract infection. And from pigs, it gets into humans, also as upper respiratory tract infection. Usually, that step here, from pigs to humans, results in the pandemic. Okay? Now, you all know that flu can be transmitted from person to person. Okay? That transmission is an example of seasonal influenza. You also know that people can acquire infection directly from birds, not necessarily waterfowl. It can be chickens, can be turkeys. So this is a non-communicable influenza. Now the question is why people cannot transmit the bird flu. Okay? The answer is in the receptors. So the receptor in birds is alpha-2,3 sialic acid. Please do not memorize these words. Receptor in a human upper respiratory tract is alpha-2,6 sialic acid. Now look at this. This is the receptor for the bird flu. This is the receptor for the human flu. Receptors are different. Therefore, bird flu cannot infect humans. Pigs have both receptors, okay? That makes sense. Pigs have both. Birds in the digestive system have alpha-2,3, and humans have alpha-2,6. Are we clear? But in the human lower respiratory tract, the receptor that we have there is also alpha-2,3 sialic acid. So, if you are exposed to a bird flu cannot infect traditionally your upper respiratory tract like usual human flu does. It can infect your lower respiratory tract, your bronchi, your lungs, which results in much more severe infection, okay, which is not really a communicable disease. Because again, when somebody coughs during the bird flu, infection, really another person does not like immediately inhales it deep in the lungs. Does that make sense? So basically, bird flu cannot infect humans effectively because receptors are different in the upper and lower respiratory tract. Now, do we have a vaccine? We do. Do we have treatments? Yes, but both vaccine and treatments are pretty lousy. Vaccine has a relatively low efficiency compared to other vaccines, and treatments have to be administered very early during the infection, otherwise they are not effective. And on top of that, this virus is changing rapidly, and we'll talk about it. Now, should you get a vaccine? Or absolutely, because it reduces the community spread. It reduces your chances of dying from severe infection, and it reduces the risk for other vulnerable populations. So in this case, vaccine would be more and more efficient if the vaccine coverage will be higher. Any questions? Now, let me explain. Look, I'm not going to talk about symptoms, okay? You all know how flu manifests itself, so like you all had it. So let's talk about the changes. Why this virus is so such changes so rapidly. So it has two phenomena: antigenic drift, which is slow accumulation of mutations in the viral RNA. RNA synthesis is very error prone, and every time RNA is being copied, mutations accumulate. We good so far? Now, another phenomenon is antigenic shift. Which is basically reassortment of genomic fragments, okay? So remember I mentioned that influenza has segmented genome. Now I'm going to use, by the way, I forgot to mention, I don't know what the view for WebEx you're using, but if you move your mouse cursor to the upper right corner of the WebEx screen, there are different views, and if you choose presenter's view, this board will be on the big screen and everybody else will be on the like green, okay? Just letting you know. So imagine that Mary has 8 different playing cards of the same type, like 8 spades. And I have 8 diamonds, okay? So we put our stacks together, we shuffle them, and then we deal 8 cards to each other, okay? So 8 for Mary and 8 for me. Now, she had all spades, I had all diamonds. When we shuffled and deal them back, now we both have mixture. Does that make sense? When 2 different influenza viruses infect a pig, they can exchange, reassort their genomic fragments. And that may result in the dramatic change in the antigenic properties of influenza virus. For instance, the famous H1N1 swine flu pandemic in 2009 resulted from the genetic reassortment between, I believe, 2 bird viruses and 1 pig virus, but I wouldn't take my words to the bank. It was definitely reassortment product. Questions? We good? Okay. Let's move on. So the next group of viruses that I wanted to go over quickly, just make sure that I cover. Yes, okay. So it's going to be flaviviruses. The name flavi comes from the Latin word for yellow, because it was named after yellow fever. So those are positive, single-stranded RNA, non-segmented, viruses with the envelope. I'm going to divide them into three main categories. First group of viruses are the viruses that cause encephalitis. And these include West Nile virus, Japanese encephalitis virus, tick-borne encephalitis virus. Okay. Second group are hemorrhagic fevers. And these include, and look, it is none of these lists is a complete list. There are many more. I talk about the most common. They include dengue and yellow fever virus. Okay. And the third group that I'm going to put here is just fevers. I'm going to put just one there because it's famous, Zika virus. Okay. So first, let's talk about the transmission routes. So dengue, yellow fever, Japanese encephalitis, and West Nile can all be transmitted by mosquitoes. Zika as well. So these are all mosquito transmitted. Tick-borne encephalitis, as you can guess, is transmitted by ticks. Okay. Now, I'm going to highlight viruses that have a capacity of human-to-human transmission. I'm running out of colors, quite frankly. So I'm going to put human-to-human transmission. And that's going to be dengue, yellow fever, and Zika. These are zoonotic viruses. Sorry. West Nile can be transmitted from birds, and Japanese encephalitis and tick-borne encephalitis are transmitted from rodents. Okay? Finally, which ones have a vaccine? So far, for humans, and I want to highlight it, for humans, we have a vaccine against yellow fever. Okay? We have a vaccine against yellow fever, vaccine against tick-borne encephalitis, and vaccine against Japanese encephalitis. Okay, so those three are the ones we can vaccinate people against. Finally, I want to review in some more details the clinical course for these infections here. So these infections are biphasic disease. What does that mean? It starts with the flu-like symptoms, which can be very severe: myalgias, arthralgias, headaches, vomiting, and in most people, that's it. but in some, it progresses to encephalitis or hemorrhagic fever. And I want to make it very clear, look, if we're talking about these three, after the first phase, some patients may end up with encephalitis, with a brain infection. We talk about these two, after the first phase, some patients may end up with a hemorrhagic fever. But it's not like going back and forth. It's not dengue can cause either of them. No, dengue causes hemorrhagic fever, West Nile causes encephalitis. Okay, there's no treatment, there's no specific treatment. Now, what about Zika? In adults, it's just a fever. In newborns, intrauterine infection via placenta leads to microcephaly in newborns and basically developmental abnormalities. Does that make sense? We good so far? Okay. Let's talk about 3 more viruses and I'm going to call it a day. So our first one will be poliovirus. It's the positive single-stranded RNA virus without an envelope, which explains why it is so stable. It is transmitted via fecal-oral route, which leads to intestinal infection. In approximately 1 out of 200 patients, neurotropic infection of ventral horns in the spinal cord. Now, ventral horns of the spinal cord contain neurons that are responsible for controlling your skeletal muscles. And these neurons are destroyed. That leads to flaccid paralysis. Usually, it affects lower limbs, but it can be as high as cervical region of the spinal cord and may affect the function of respiratory muscles, which can produce, some very unwanted effects. All of them are unwanted, but people actually lose the capacity to independently breathe. Okay? So far, we good? Now, what I want to make absolutely 100% clear is that as far as it's a human virus, so only human. So if we vaccinate each and every person in the world, we can eliminate polio. And there are 2 vaccines. One is inactivated polio vaccine, developed by Jonas Salk, okay? And it has some advantages and disadvantages. Advantages, it's very safe and effective. Disadvantage, it is injectable. And does not provide mucosal immunity. So all of you have received this vaccine, an activated Salk vaccine. So what would happen to you if you get exposed to a poliovirus? Will you be infected? Surprisingly, the answer is yes. The virus will reproduce in your gut, but you will be protected from the disease. You will not develop polio. Do you see the difference? Makes sense, right? Now, another vaccine, which is oral attenuated vaccine, developed by Albert Sabin. It contains the replicating virus, which is weak. So the positive oral administration, you can imagine, injection has to be done by a trained professional. You can give an oral vaccine. I can do it. Everybody can do it, okay? Just give a drop. And it provides a robust mucosal immunity. I mean, massive amount of immunoglobulin A in your gastrointestinal mucosa. Negatives. It can cause so-called vaccine-associated polio. When one of the strains gets in one of the vaccine viruses inside of a human intestine, can revert to a wild type, and this wild type virus can cause a vaccine-associated polio. The frequency is pretty low, approximately 1 per million vaccinated, but still. Okay? There's no specific treatment, by the way. There are a few countries that are still not polio-free, like Afghanistan and Pakistan. Other than that, as far as I know, the rest of the world is considered now to be polio-free. We good? Can we move on? Mary? It's kind of, I can see your eyes, how you track what's on the screen. This is why I ask, you got it? Okay. Leon, we good? Okay. We have 2 more positive single-stranded viruses to discuss. Hepatitis C. So it's another flavivirus, but it's slightly different. Single-stranded RNA virus, which is enveloped. And hepatitis C, positive agent of, well, hepatitis, the liver infection, which may result in cirrhosis, which may result in the liver cancer. We'll get back to that clinical course. Hepatitis C is entirely human infection. Main route of transmission. It's a blood-borne pathogen. So any exposure to the infected blood, like a needle stick, or sharing the needles between the people who use intravenous drugs. Sexual transmission is minimal. Vertical, from mother to child, is also possible. Okay? Now, one of the big problems with hepatitis C is that there is no good animal model. One may ask, what about monkeys? Monkeys are extremely expensive to work with. And there's a lot of pushback from using monkeys as the animal model. There is no good cell culture model. So this virus is actually quite inconvenient to work with, okay? And on top of that, there's no vaccine. However, there are treatments, are very effective. In fact, the course of studying this virus led to the development of extremely effective antivirals that can cure up to 99.5% of people infected. Okay, so virus is undetectable. Those treatments are expensive. I take this, but they are extremely effective. There was something I wanted to highlight, and I totally forgot what I wanted to. Oh, yes. So if you will look at the number of people infected with hepatitis C, it turns out that there's going to be a fraction of people that will clear the virus by themselves, a fraction of people that will develop chronic liver infection, and nothing's going to happen, really, like just a liver infection, liver inflammation. In some of them, it will progress to cirrhosis, and in another fraction of those with cirrhosis, it will progress to liver cancer. So not everybody gets the liver cancer. Okay, we good? All right. Any questions on Hep C? The last virus that we're going to talk about is one of my favorites sorts, and it's also one that Ohio can be proud of. It's the norovirus, which is positive, single-stranded RNA genome, and it's naked virus. So this is an entirely human infection. It's not zoonotic. Transmitted by a fecal-oral route. Now, when I say fecal-oral, and it causes gastroenteritis. It is the most common cause of gastroenteritis in the infectious gastroenteritis in the United States. Virus is extremely contagious. The median infectious dose is low. So even 10 particles are enough to establish the infection. The virus, the course of disease is pretty mild. It's basically, sometimes it's called winter vomiting bug. Sometimes it is called a two-bucket disease because patients basically experience diarrhea and vomiting. And it lasts for about 24 to 48 hours. Then they recover. Now, what's fascinating about it, it can be shed during incubation, disease, and convalescent stages. During all those stages, the virus can be shed. So even if you recover, you can still shed the virus for up to 2 weeks. Okay? It is often transmission, since it's a fecal-oral route, transmission is mostly in the community, schools, nursing homes. It is famous for causing outbreaks on cruise ships. Contaminated food is usually the culprit. Somebody who had this infection before contaminates the food, and here we go. It is spread like a wildfire. I want to mention this. Let me check something out. I want to mention this. There is no robust immunity, no vaccine. So it turns out, when you get infected with norovirus, you can be reinfected with exactly the same strain about 6 months later. And you can be infected with a different strain about 2 months after you recover from original infection. So we don't know why there is no robust immunity. We do know that there is none. Okay, good? Any questions? That'll conclude the viruses.